If you were in the last session, you already know, but I'll reintroduce myself. My name's Adam Kaufman. I've been a board member at the CMTRF since 2019. I'm also a patient. This next session in this room is titled CMT Type 1, Mechanisms and Planned Interventions. Our speaker again is Dr. Mary Reilly. She's a professor of neurology at the University College of London, Department of Neuromuscular Diseases. And I'd like to welcome her to the stage. Thank you.
Thank you very much. So for those of you that didn't meet me a few minutes ago, I'm going to continue where I left off on CMT1. So as we said earlier, CMT stands for Charcot-Marie Tooth disease, and the basic definition of the classification is some forms of it, the problem starts in the myelin and it's called demyelinating. Some forms it starts in the axon, it's called axonal. And traditionally, we call CMT1 when it's demyelinating and CMT2 when it's axonal. But I want to stress again that if it starts in the myelin, the axon eventually is affected and vice versa. So that's really important because particularly in CMT1A, the disability is proportional to the damage to the axon, not to the damage to the myelin. But in terms of treatments, that's the way we think of them. So we talked about the classification, and CMT1 is autosomal dominant, and there's a number of genes. And as we said, the letter refers to the underlying gene. CMT4 is autosomal recessive, and CMT2 is the axonal. And then there's the X-linked. And the ones I'm going to talk about today, looking at what else has been covered, I'm going to cover the autosomal dominant CMT1, but I'm also going to cover briefly X-linked because it doesn't fit into one of the other rooms. Now, there's a relatively short list of genes for CMT1 autosomal dominant compared to, say, CMT2. So CMT1 is commoner, but there's less genes that cause it. So most UK, US, Northern European populations, about 50% of people have CMT1A. So it is the commonest versus the others. And the second commonest is JGB1. And when we looked at our cases in the UK, if you take those three genes, three commonest genes, PMP22, MP0, and JGB1, it accounts for about 66% of our cases. So if you look at our series of patients, out of 1,700 patients, 43% had CMT1A due to the PMP22 duplication. CMT1B is reasonably rare, 3.3%, and X-linked JGB1 is 11.9%, and all of the others are extremely rare, less than 1% or 1%, so they're very rare. Now, why is 43% a little bit lower? It should be about 50. That's because if you run a very specialized clinic, some people don't come to you if they're already solved. You tend to have a bias towards getting people that don't have a diagnosis. So in specialized clinics, we often see more of the rarer types and less of the common types, but still the common type is the commonest. So they're the ones I'm going to mainly focus on. I just don't have time to go through all of these. It's not saying they're not important, and they're very important. Every CMT is important, but just covering the main ones. So CMT1, we're talking about disease starting in the myelin sheath that surrounds the axon. Now, what does the myelin sheath do? So if you look at a picture of a nerve, so that is the axon, and that's the myelin sheath. So the myelin is for conducting the impulse from the spinal cord to the muscle and from the skin up to the spinal cord. Now, to conduct an impulse, you need to have very good quality myelin. So the myelin needs to be in good shape, the right thickness, and to have no damage. But as well as that, if you look at it, there's gaps along a nerve, and these are called the nodes of Ranvier. So every little bit of a distance, there's a gap. And when you have an electrical impulse, it doesn't just go like this, it jumps from gap to gap. So you need both normal myelin and you need normal nodes of Ranvier. That's called saltatory conduction. So you need all of that to be working to have normal myelination and normal conduction. And that's what the node looks like. So when we think about working out how to treat CMT, we start with the patient. The first job is to find the causative gene. The next job is to find out how that gene causes the disease. So that's often where we do a lot of preclinical work. Find out what that protein that gene makes cause the disease. Once that's done, we try to develop candidate therapies. They go through preclinical trials, eventually to a clinical trial, and for a treatment back to the patient. Now, I call that circle of translation. Now, we're very good at this end of it. We haven't quite got to this end of it yet, but we're there. We're getting there with clinical trials. But that's what we're talking about. We're talking about translating. So in terms of therapies, I'm going to just dwell on this for a moment. I think of them very simplistically. So everybody has a code of DNA that gets transcribed to messenger RNA and translated into a protein. So when you develop treatments, you can do three things. You can do what's been done traditionally, work out how the abnormal protein causes the disease, and you have what's called pathogenetic therapy. The therapy is based on what the protein does wrong. You can have gene therapies, which are really what's very exciting. It's almost like it doesn't matter what disease you have, you can develop therapies that work at the RNA level or at the DNA level. Or you can have pathway therapies. Doesn't matter what type of CMT, have we therapies that can treat nerves? Imagine if we therapies that could treat nerves and it didn't matter what type you had. So in gene therapies, I just thought I'd use a few words to get them out there. So you'll have heard of antisense and silencing RNA. Those are therapies that can affect the messenger RNA and change it so that it either makes a normal protein or changes it in some way. And then if you go back further to the DNA, you can edit the DNA permanently. There's ways of doing that. Or you can give a replacement gene with viral. Now, the thing to say, if you do these two things with the DNA, they're permanent. It's a one-off treatment for life. And that's when you hear about gene therapy, replacement therapies. But when you do these ones, which are in practice now, we have them for other diseases, they need to be repeated. So they need to be given and repeated. And there's advantages and disadvantages to both. You know, when you're talking about DNA, there's advantages to doing it and you can reverse it and to doing it permanently. And that's something that's been looked at. So they're just some of the words you'll hear used. And people use gene therapy to account for just this bit. Sometimes they use it to account for it all. I think it just covers everything. And then you go into the individual bit. But sometimes it's confusing for researchers, clinicians, and patients what people mean. Now, if we look at the commonest types, so CMT1A, we've known about the gene since 1991 and we still don't have a treatment. Now, what does it do? Well, you find it in myelin, but it actually only accounts for 2 to 5% of the myelin. So even though it's a very small protein in the myelin, it's critically important. And actually, we don't quite understand what it does, but we do know how it causes the disease. Now, the history is very typical. Patients have onset in the first 2 decades. And I would say it's always in the first decade if you look back. Because they may say they come to the doctor at 12, 13, and you'll say, well, what would you like? And my standard question is, if you're 8 and you were doing a 100-yard race, where would you come? And it's nearly always pretty near the back. It's not always, or they're clumsier. So it's often early in life they get it. But it's a slowly progressive condition that affects the feet first, then the hands. It's length dependent, so it's always the feet first and then the hands, although I would say towards middle age, the hands are a huge problem for patients. They've got used to the feet, but they find the increasing hand weakness can become quite troublesome. Foot deformity is a huge problem with CMT1A because there's unequal involvement of the ankle muscles. That's a huge problem for it. And often it's very rare to need a wheelchair full time. It can happen in rare patients, but often people need aids to walk when they're older, be it orthosis, be it a stick or a cane, you call it here, or some sort of frame. And, sometimes, patients, and I think men particularly, I hope this isn't sexist in any way, really resist using aids. Now, the word wheelchair to a man in clinic sometimes is like the wrong word. But if you go to an American airport, you might sometimes have to walk 3 miles, as far as I can see, when they call your plane to where you have to go. So sometimes people use transport for those kind of things. They don't need a wheelchair, but they will use it when they need to. And that's sensible. Conserve your energy for having the drink on the plane or whatever. Just pick your battles. Now, chromosome 17 is the gene where the PMP22 protein is on. So it was the first one to be described. And what you get is you get an unequal crossing over. So basically, the PMP22 gene is on chromosome 17. And what happens is you get a small bit of the chromosome that duplicates. It folds on itself so that this person here has 2 copies of the gene rather than 1. But if that happens, some people have only 1 copy of the gene. So remember, you'll have got one of the chromosomes from your mum and one from your dad. So it's a dosage problem. If you have 2 copies of the PMP22 gene, one from your mum and one from your dad, you're normal. If you have 3 copies, you have CMT1A. And if you've only one copy, you've hereditary neuropathy with liability to pressure palsy. And I'm just going to mention this because it tends to get forgotten because people think it's a less severe disease. This is where your nerves at points of pressure, such as your elbow, your wrist, or when you cross your legs, are liable to pressure palsies. Now, if I fall asleep on an airplane for a 14-hour flight like this, I'm likely to wake up with a problem with my hand being a dead arm. But if you have this condition, that will happen very easily. And what people find very distressing about it is it's unpredictable. So with CMT1A, by and large, you know your symptoms. But with this condition, you don't. So I had a patient who was a nurse who decided, for some reason, to get half her body tattooed. As I said, interesting decision, but it meant she was lying in this position for about an hour and a half. And she got a paralysis of her arm. Now, it came back eventually, but that's what you can't do. So that is what hereditary neuropathy with liability to pressure palsy is. So we know from gene dosage studies, from cell cultures, from rat and animal models, and from humans that it is dosage. So 2 copies, you're normal, 3 copies of CMT1A, and there are rare patients who have 4 copies, and they have more severe form of CMT. And I think this is the proof. This is an absolute proof. There are 2 families in the world like this. So one of the parents has CMT1A. So they have 2 copies of PMP22 on one chromosome and 1 copy on the other. So they have 3 copies. Everyone follow that? And then their partner has HMPP. So they've only 1 copy of PMP22. They've 0 on one chromosome and 1 on the other. And one of their children inherited the 2 copies from one parent and the 0 copy from the other parent. And they had 2 copies. And although the 2 copies came from one parent, they're completely normal. So it doesn't matter where you get the copy of the gene from, you can be completely normal. So we know the cause of CMT1A is too much PMP22. And I have this very rare family who is another very unusual problem that happens to PMP22. And that's CMT1A, that's the PMP22 level, that's normal, and that's HMPP, and that's my patient. And they have a much more severe form. So we've loads of evidence that what we need to do is reduce the amount of PMP22. Now, as you will see from this picture of the current therapies, there's been more therapies developed for CMT1A than any other. And many of these didn't get into clinic, but because it's the commonest and this the original gene, most work has been done on this. And the first trial was the ascorbic acid, that's vitamin C trials many years ago, and they did not work in multiple trials all around the world. And saying that, we learned a huge amount. So it wasn't in any way a waste of time for people. We learned a huge amount. Then there was Pharynx. And I'm just going to mention this because many of you will have heard of this drug. So this was a drug that was developed by the company predicting by computer analysis what might reduce the PMP22. And they came up with this drug with an opioid, GABA receptor, and sorbitol. And initially, the animal study was positive. The phase 2 study was hopeful, but the outcome measures they were using were not great. And that wasn't their fault. It's what the FDA was saying they had to use. They did a phase 3 study that looked hopeful. But during the phase 3 study, the high dose crystallized out. And if you're getting something and it crystallizes, you'll know you're not on the placebo. So the FDA, quite rightly, said they had to do another phase 3 trial. That ended in December 23, and they reported publicly that it was negative, but there hasn't been any papers published since then. So it looks like that study was negative. There's another one they were doing in partnership in China. We haven't heard about that. So it looks like Pharynx, the report is that it's negative, but as I said, we haven't seen the final reports of that. But this is a disease waiting for gene therapy because what you want to do is reduce the amount of PMP22. And these things here, the antisense and the siRNA, are genetic ways of reducing the expression of a protein in a way that can be reversed. It's something that can be done and then reversed. And basically, as I said, this would be a perfect thing to do. So if you think about it, you want to reduce it by about a third. So you want to get it down to the equivalent of 2 copies and you have 3 copies. So you're trying to get a drug that will reduce it by about a third. And the first animal model of this was done in 2018 and it was very successful. So proof of principle that if you do reduce it, you can stop the disease progressing. And in this case, it improved a bit. And this is very exciting because multiple companies have now taken this forward and developed drugs. And probably the one that's nearest is the Falcon one here, which was developed by DTX, and Novartis have bought it. Many of you will have heard of that. So they have a product which we hear will come to trial in 2026. It's not quite, hasn't happened yet, but we hear it's coming to trial. There's another French one that's very near it as well, and there's multiple other ones. So I do think it is very likely that there will be a clinical trial of one of these drugs in the next year, and it's likely to be intravenous. That means given by an injection to your veins. Now, in terms of just to mention that there are what we call pathway therapies. There's also another drug that's in trial, which is a drug that's been developed looking at the neuromuscular junction. Now, again, if you look at a nerve going to the muscle, there is this thing called the junction. So when the impulse comes down the nerve to make the muscle move, there has to be a connection. And a group of people looked at both animal models and humans and worked out that in multiple forms of CMT, this neuromuscular junction is important. And if you inhibit a channel called a chloride channel, you can improve function. And this has led to a clinical trial that's now ongoing in multiple forms of CMT, and it's happening in the US and in Europe. So that's something to look at muscle function across different forms of CMT. So that's one that's also will be patients with CMT1A involved in. The second form of CMT is CMT1B. This actually, and I was thinking about this because one of the patients here has 1B and asked me what's happening. And I kind of dwell on this. We haven't done so well with 1B. And actually, it's something we should do more on. And 1B is really important. 50% of myelin is myelin protein 0. So it's an extremely important protein, critical to have the right amount of 1B. And patients can get a variety. They can get a very early onset, severe form of CMT, and they're very sensory ataxic. That is, they're very unsteady. And to me, they have a lot of neuropathic pain. Or they can get a late onset variety, which is milder and comes on older in life. So there's a wide spectrum. And it's a lot of work done on it. But what seems to happen is it's called the unfolded protein response. Now, what I mean by that is, if you have proteins that misfold, so they don't fold properly, they go to a part of the cell called the endoplasmic reticulum, the ER, and that has a very efficient way of getting rid of them. So this protein 0 doesn't fold properly, but it overloads the ER. So the ER can't cope with the amount of unfolded protein, and therefore it accumulates. And basically, a drug has been developed called Cephin-1, which helps this pathway work more efficiently. But that's a very attractive treatment. And in an animal model of CMT1B, it works. But as yet, for various reasons, it hasn't been taken to clinical trial in CMT, but there is an ongoing trial with it in ALS. Now, one of the issues, that I'm concerned about is this pathway of unfolded protein response is really important in every cell in our body. So you'd have to be really sure there was no side effects for this. But actually, there hasn't been much else developed for CMT1B. Genetic therapies, there is a complication that would need to happen with that, but it is possible. So I think that's an area we have to think about carefully because we haven't done as well as other forms of CMT. And again, pathogenesis therapies can happen there. And the final form of CMT I was just going to mention was X-linked CMT. So I mentioned this is due to connexin 32, JGB1, gap junction B21 is the gene, and connexin 32 is the protein. And these are found in those important nodes of Ranvier. So remember I said the conduction had to jump. That's where you find connexin 32. So it forms these channels. That's what it looks like. And these channels, you can see it here, are near the node of Ranvier. And what they do, they're very clever. These channels connect the axon to the myelin, to the Schwann cells. These are really important connections. Maybe that's why it's intermediate. It's connection between the axon and the myelin. So really, really important. Now, we know from studies, including from many people in the room here, that what happens with X-linked CMT due to JGB1 is a loss of function. It just doesn't work properly. So what we should be doing is doing gene therapy, replacing the gene. And that is what has been done in animal models of the disease. And this has been work, quite a lot of it led by Kleopas Kleopi in Cyprus. So he has successfully treated a couple of different animal models of CMT successfully. But one of the problems is translating that to humans. There's a lot of questions yet to be asked in terms of safety, in terms of how you'd give it, etc. And finally, there are challenges with developing drugs for CMT1. And the one I'm going to mention today really is delivery. So how do you get the drug to the nerve? So do you give it through, what we call a lumbar puncture and hope it'll get down. And if you give it by tablets or by injections, you want it to get into the nerve, and there is what's called a blood-nerve barrier. It just won't float into the nerve from the blood. You have to have a barrier there. And you want to give it in doses that are small enough that it won't cause problems with the liver. Now, that may all sound very, things we should be able to deal with, but they are the biggest problem at the moment in developing therapies. We all say is delivery, delivery, delivery. We have the drugs. We know how they work. We know they do work, but we have to work out how to give them. And safety is huge. CMT1A, patients with it have a normal lifespan, as do most forms of CMT. So we do not want to be given a drug that has high risks with it. It has to be safe. So that's one of the reasons with CMT, it may seem slow to patients, but the safety barrier is huge. We have to. And maybe because I'm in the US, I use the analogy, it has to be as safe as a packet of M&Ms. It has to be completely safe before it justifies giving it. So I hope I've given you a flavour for the developments in CMT1 and the underlying reasons why people need therapies. And again, just to thank my group in Queen Square who work on CMT, and I'm happy to take any questions. It's a bit of a fast run through the common forms, and I appreciate I won't have covered any one in detail or many at all. Thank you.
Hey, Mary, lovely talk. In terms of pediatric considerations, what do you say to parents who have children with CMT1A or who may be going through gene therapies? Like, will it be tested on adults first, or?
So I think that's a very good question. So I would say it depends on the type.
Yeah.
With 1A, the feeling is very definitely from everyone, including the regulators, it has to be done in adults first because for safety reasons. But we do know that we really would want to be treating in childhood before there's axonal damage. But I think safety reasons, and adults, in the UK, adult is defined as over 16 for clinical trials. In the US, it's over 18. And, people can be very mildly affected then. So I think if we were designing trials, we'd want to make sure there was some younger people in them. For other forms that are much more severe, clearly the treatment initially should be in children. And there are trials being developed for gene replacement therapy for CMT4, which will be in children. So I think, when we're talking to regulators about what we're allowed to do, it depends on the severity and the safety. So it's a different answer for each type. I would say CMTX will also be adults first. CMT1B, it probably will depend on the therapy. Because sometimes, people can be very severely affected early on. Sometimes they're not. So it'll depend.
And then one more question. Can you talk about accessibility to these gene therapies, whether you're in the US market or the European market?
Sure. Well, that's an interesting, tricky question. So first of all, accessibility to clinical trials. Clinical trials will happen where there are good setups to do them. So you have to have a setup which is used to seeing patients, which is used to doing clinical trials. And then there's a question of people being able to get to it. So, companies, if it's a very rare form, can fly people in from all over the world. And there's trials going on now that are multinational trials. And as a community that work in CMT, we work internationally. We've always worked internationally because we know that's where the answers is. And for the rare forms, is how we work it. Now, when it comes to therapies being developed, and I've experience of this for amyloid, TTR amyloidosis, these therapies are expensive. These are not cheap therapies. And the reimbursement varies in different countries. Drug companies want to license in the States first. I don't know, should I be saying this, but drug companies do because the States pays more money. So overall, and also in the States, most prescribers, they can prescribe it. So in the UK and Europe, often it's a national decision. So we can prescribe a drug. It nationally gets decided whether we can or not an individual. So they're different. So it's a different issue. So I think in most, if you want to use the term first world, but developed countries, they will get licensed, even if they're expensive, if they work. Although I would say with TTR amyloid, New Zealand hasn't licensed it yet. 6-7 years after the papers, you can't get it in New Zealand. You can get it in Taiwan. You can get it in Brazil, but you can't get it in New Zealand. So there are exceptions. But there's a huge inequity around the world. There's a huge problem with equity. And I just hope, and I may be wrong, that, the technology behind developing these, I'm hoping other companies in these countries will be able to develop lookalikes, depends on the patents, of course, or that companies will find a way of making it that it's available. But in certain parts of the world, and we get emails from patients, you know, they're not going to get a diagnosis in some of these places yet. So there is inequity, and it's a huge problem, as it was for HIV treatment, as it is for cancer treatment. So it's not unique because it's CMT. But I think, you know, there are people being able to get to clinical trials and able to get therapies is going to be really important. And somewhere or the other, this being affordable and being seen. And I think that's why, certainly in the UK, what we have to do is just constantly ask our patients to advocate that this is a serious illness, that there needs to be a therapy, that it's an unmet need. You know, we need to do that, and we do that now. We don't wait till we have the therapy. We do it now. So we're building up to that. Thank you.